And greetings, everybody. Welcome to the Evidence and Reasons for the Christian Faith video channel. I'm Salvador Cordova. I'm joined by Rebekah and Michlas. And if you're wondering why there are two Sals on the channel, I'm having some technical difficulties where one computer has to provide the audio and then my smartphone provides the video. Backstage, we were running this and the audio, I was told, was just terrible. So this is a workaround. I want to thank you all for your forbearance in advance. So this meeting is was requested by Michlas just to talk to me and Rebekah about a variety of things. I'm just saying this is shooting the breeze. Uh, Michlas is on the other side of the Atlantic. I'm on the east coast of the United States, and Rebekah is in the mountain time range. So it's really difficult for all three of us to be able to have a meeting like this. It's great. So uh, I'll have Rebekah introduce herself briefly. Most of you know who she is, and then Michlas.
Hi, everyone. Well, if you don't know me, I have a channel called Bread of Life. It's my biggest channel where I talk about Jesus and topics related to faith. And then I have another channel called Examining Evolution. And Michlas, Danish debater, came on my channel pretty recently to discuss ERVs. And we had a great conversation. So this conversation is kind of like us responding, me and Sal responding to some of the prediction that Michlas made and some of his observations with regard to the discussions on ERVs that I had with Dr. Dan and Donnie.
So yeah, and as they said, my name is Michlas, or Danish Debater Online. I do not have a channel, or I do, but it's not one of importance. I have no interest in growing an audience. What's interesting to me is communication between people, how we best settle our differences and actually reach a point where we can actually talk about what we disagree about. And that's one of the reasons that I have been on Rebekah's channel and why I like Sal, among other people, because while I am on the opposing side to their opinion about there being a God on young Earth and on evolution, I actually respect them and like them because I think they do a good job in communicating their views. And well, I was on Rebekah's side talking about ERVs, where I tried to give my best view as a layman on presentations that have been on her channel, and then why I still found the evolutionary side of it more convincing than the young Earth creationist side. And I would like to hear a bit about what you thought about that.
So we agreed that Rebekah and I are at Michlas's service. He asks questions, he directs the conversation, and so we are privileged here to be of service to you, Michlas. And both Rebekah and I think very, very, very highly of you. And we really did look forward to seeing you and interacting with you today. So just fire away, brother.
Well, thanks a lot. Now, Rebekah said she had it in recent memory. So maybe if you would start with just first impressions and maybe questions you would have for Sal regarding some things I said, or for me for the matter.
Yeah, so, well, I don't know how beneficial this is for the audience, but if they haven't seen this video, but I will say that when Michlas presented the information about ERVs and made observations, he was very fair to both sides. So I wasn't expecting that. And so I felt like he did a good job of you did a good job, Michlas, of really understanding the young earth creation side and being willing to concede points that I really don't see being conceded on the other, from the evolutionary perspective. I don't see them often willing to admit when young earth creationists have made a good point. And so I think one thing in particular that you mentioned was, I said, well, how do we know that these are not original features, and that they've just been labeled endogenous retroviruses? And I said, and you agreed with that. You said, well, we don't, but here's some things that we would expect. And I think the main point of your presentation, I thought it was really excellent that you went through working, like you kind of went through an exercise working out what we would expect to see if young Earth creation is true, and what we would expect to see if evolution is true. So with regard to like what we would see in the ERV mutations, and this, Sal, so Michlas made a prediction, Sal, and what neither one of us really know is if there's any observations that would confirm this prediction. And so that's kind of what I'm most interested to find out, is if there actually are scientific observations that we can point to say whether this prediction is like which side it kind of favors. Okay. So here's the point, the prediction that Michlas was making. If young Earth creationism is true, then what we would expect to find in shared ERVs, okay, like ERVs that are shared among many or all organisms, right? If there are shared one, I don't know if there's any that are shared among all, but shared among many, okay, then we would expect that those would have more mutations than ERVs that are wait a minute. Oh my gosh, I'm so confusing myself. I can't remember the point I was making. Hold on. I can explain this, okay?
No,
But
go ahead. I know where you're headed. I could probably explain it better, but why don't you give it a shot and then I'll give it a shot. Is that all right with you?
Okay, I will explain it again. But I'll do better at not losing track of my thoughts. And then Michlas can correct me if I'm saying anything wrong. Okay, so the issue is that we have ERVs that if they were originally there, like if they were originally designed and part of the organisms, then we would expect that we would see more mutations in them because the mutation, there would have been more time for mutations to take place within those, right? But if those are maybe the reason I'm getting confused is because I'm talking about the farther back ones, but I should be talking about the closer ones. Michlas, you're going to have to jump in here.
So yeah, it was pretty close. So I said,
Okay.
if we have evolution where the ERVs has come in over time, then we should expect that those that most animal shares came in first and thus have most mutations.
Yes.
Where if it was created mutation, or not mutations, ERVs, such as the idea where it's made by God as a defense against later viruses or other things that were in the original videos, then we would expect that they have all had the same amount of time to accumulate viruses, so the distribution should be the same. That was one of the predictions. The other prediction was that if we take only the mutations, and only that, not the ERVs themselves, but the mutations within the ERVs that we agree are broken. That's pretty important that we have to agree on both sides that these ERVs are broken, and we take the mutations in there, then we should see the same pattern as before, that the earlier they were broken, the more mutations, and that would be on both young Earth and on evolution, because when they are broken, they are no longer experiencing purifying selection, so they would both experience the same amount of mutations. But those mutations would generate a phylogenetic tree under evolution, but they would not do that under young Earth creationism. Does that make sense?
We might have to work through this slowly. And people wonder why sometimes I don't engage in certain discussions like ERVs. It's because I feel that we have to wait a little bit. There is a lot we do not know. And if I could just I need to take 2 side trails to explain where I'm going, and hopefully it'll enlighten. In 2016, Dr. Sanford and I were in a private conference, and he said, of anything that would make me become an evolutionist again, it'd be the ALU elements. ALUs are transposons, and ERVs are transposons. So it's a comparable problem. And right after the meeting, he said, Sal, why don't you look into this? I'm just like, I don't think I have a chance. This really does look pretty bad. If you think that this is such good evidence for evolution, you're going to ask me to work on this problem. And I said, okay, well, you're the boss. I'll go ahead and do it, sir. Yes, sir. And I found out, in 2009, if he had assigned me this problem, I would have no chance. Just a year later, 2010, the studies came out. 2016, there were even more. And this got incorporated into the book Contested Bones. So, in 2010, Francisco Ayala, all these guys were saying ALU elements, these repetitive transposons, are just absolute junk. The designer's totally incompetent. This is consistent with random evolution. By 2016, it's like, no, this is some of the most incredible computational, since you're in computers, Michlas, this is like random access memory that gets modified. That's why they're all identical. They can be modified on the fly. And I said, we were so committed to a particular way that we thought DNA worked. And we found out chemically, it is, when it expressed as an RNA, it can be modified like a random access memory. No one saw that coming. Absolutely no one saw that coming. And I'm just like, gee, 40 years ago, this would have seemed like a slam dunk case, enough so that someone like John Sanford, who is a young Earth creationist now, said this would make me go back to being an evolutionist, because this superficially looks like evolution. But then experimentally in the lab, it got overturned. Well, 2 years later, he said, Sal, that was a good job on the ALU elements. I want you now to do research on the ERVs. I'm just like, what am I going to do? And he wasn't expecting me to do the lab results. He said, just look at the literature. We have Google, look in the libraries, find what you can. And I'm like, Lord, okay. I was just like, no way. Well, just even that year, there were a few breakthroughs. And even now, there's been a breakthrough 4 years later. So that's one. Let me just say, this is like one rabbit trail. Just saying, we still are very early on in determining what really is there, how it works, and its role. Things that we might say are broken or whatever, we still don't know. There are broken parts, and I will. Okay, now the second point. Now I remember it. The thing with geocentrism, they're able to make tremendous predictions, like the sun rising every day. They could predict eclipses. They could predict retrograde motion using epicycles. It did mean that the model was fundamentally correct. And so I can even concede that it's consistent with some evolutionary observations. I love showing like the picture of the bent pencil or geocentrism and say, you can come up with a theory that's fundamentally wrong, but and it can also make testable predictions. So you could look at the water, the pencil. My favorite's like the pencil and water, and it looks bent. Well, you can make predictions about how the pencil is going to be shaped. It's still straight, but you can still argue that Well, it bent it, and it's going to have a certain geometry. But it's a fundamentally wrong theory. Same, even more so for geocentrism. So some creationists are just so quick to say, no, that's not, that prediction failed. And it's like, actually, it may have succeeded. And it could still be a wrong theory. It could succeed and still be a wrong theory. And I've told creationists the way that you actually can break common ancestry. I said, why do you want to go into genetics? That's probably the most messy area. If you're able to show that the earth is young, then even the evolutionists will agree with you. So you have that one, I often say one ugly fact can destroy an otherwise beautiful theory. And I don't want to make this about, and this is a good point that PhD Tony in the debate brought up. He said, this isn't about atheist versus Christian. You could have Christian, young earth creationists, and then, maybe progressive creationists who are old earth, or like Michael Behe, who believe in universal common ancestry, but still believe in intelligent design. I consider some people like that as brothers in Christ. And so this is a scientific question, but it is fundamentally settled whether you can prove the earth is young. If you prove the earth is young, then you have to figure out why you have these patterns. And I've actually worked on some of that in a paper that is under review right now, and God willing will be published soon. We have good indications from the editor. So that's briefly what I could say. I could also talk about some things about ERVs. Did Dr. Dan or Donnie talk about the CAP1 complex and zinc finger proteins at all?
That sounds familiar, but it's been a while now since I had that.
Okay.
I don't think in those talks, no. He has talked about it, but not in those.
But Sal, before we go into that, can you discuss what you know about Miklas's prediction? Like, okay, a shared ERV among created kinds, the ERV would be sitting around a long time to collect mutations. Whereas, yeah.
All right. So remember, you only have 6,000, let's say if we define young earth creationism as being 10,000 years, as a matter of principle, it had to be created. There's not enough time for one creature to evolve to another, even a chimp and a human. So that's why I just say cut to the chase. And people will say, well, that's really hard to prove young earth creationism. I absolutely agree with you. And that's why I say young earth creationism is hard, intelligent design is easy. But trying to argue young earth creationism with genetics, and I study some genetics, I said you're just going to have a mess. You really need to solve the problem of radiometric dating and the things PhD Tony pointed out in my debate with him. If that's overcome, and other things, and I've had people on my channel with opposing views, I said, this is what really has to be solved. Otherwise, you're running around in circles trying to argue it with genetics. So I would just have to say, I think Michlas has a good point, and the only way to refute it is not to criticize his genetics. The way to refute it is to do, more basic physics and address things like what PhD Tony said. And I know that's not, that's why I kind of indirectly said, even geocentrism made testable predictions that were absolutely correct, but it was the wrong model of reality. But it was fabulous. It could make testable predictions that would affirm it. You have to look at the anomalies that eventually broke geocentrism, like retrograde motion, the Coriolis force, the centrifugal force at the equator, et cetera, et cetera, et cetera. And so what will break this deadlock where you have two opposing views is if there's a change, if there's advancement in the physics of the age of the Earth. It's going to have to be something outside of genetics that's really going to settle this definitively. And I know that's probably not the answer you were expecting, but that's the way I argue it. And that's why I don't delve too much into just like nitpicking on, well, Dr. Dan said this, and so and so said that, and they're experts. Dr. Dan's an expert in virology. It's like, I totally respect that, and it's like I have, the angle that will break this is the young Earth creationism. And I will quote Dapper Dinosaur of all people. I said, Dapper Dino, if the Earth is young, wouldn't you agree then that all these kinds are created? That there's no universal common ancestor and everything's more like an orchard? He said, yeah, but that's kind of hard, Sal. So I'm sorry to give you not like the best genetic analysis. In other words, I totally respect what you said, Michlas, and I'm not going to attack it directly. I think you have great insights, and I just going to leave it at that, because professionally, that's the way I deal with that problem.
But do you know, like, have we like tracked the number of mutations within shared ERVs?
Okay, so when they track the number, okay, this relates to my professional work, and I'm glad to highlight it.
Okay.
You're assuming first that they are mutations over time, and I just said if you have only 6,000 years, that's created. Those differences are created. They're not mutations. And I can understand why you'd say, okay, if you do assume that this is old, then they would be mutations. And I would absolutely agree that this would look consistent with evolutionary theory, just like, and I pointed that out. If I could show a diagram from my talk at the Creation Research Society.
Just a quick note there. You would probably not have as many mutations as evolution says are mutations. What I'm talking about is the mutations that you would have over 6,000 or 10,000 years. You would still accumulate some mutations, right?
Right.
And this is also why I focused on the so-called broken ERVs. As far as I understand the young Earth model, the ERVs were created for some purpose, and they were created correctly or perfectly or whatever you want to say. But some of them have broken throughout time. And that's why I'm focusing on them, because these should, according to my understanding of genetics, be those with the most mutations. Not to say that the others won't have mutations, but those would probably be way fewer, at least in 6,000 years.
I'd say you have a good argument there, that in 6,000 years, there is something that Dr. Dan talked about, the crossover rate. And I said, Dr. Dan, that was a pretty good argument. And both Emery and I saluted him on that and said that, we do think that is a legitimate problem.
What is the problem of the crossover rate?
Even I don't completely understand it.
Can I try to say it as I understood it? Because I did see that one.
Mm-hmm.
As I understand it, it's a problem with created heterozygosity, that the created genes in Adam and Eve would not have had time enough to cross as much as would be needed for all the diversity we see in the genes today, because what we know is that it is, if I remember correctly, about one crossover event per generation, which would not be nearly enough of what they needed. So you would also need to have some sort of basically targeted crossover selection for the diversity to arise. And I can't remember if that would also be a further problem with the flood added, because you would have another bottleneck event which would limit your genetic sample size. So not only would you have had to have a targeted selection before the flood, the wives of Noah's sons would also have to be as widely selected as they could be, basically, among those people that were alive at that point. And then afterwards, you would again need to have targeted crossover selection for all this diversity to arise.
Now,
so basically, God would have to tinker all the way through to make this diversity.
Now, the reason that I'm skeptical about this problem is because I feel like even with vastly bigger amounts of time, you still, if we're taking the evolutionary model, the types of changes you have to get are so much worse for this problem, right? Like you have to actually develop new systems and new features within these organisms in this period of time. You're talking about, differences in hair color and eye color and skin color, which are very superficial, right? I mean, compared to like the types of things that would have to happen for the evolutionary processes.
So it's two different issues. I do understand the issue that you're talking about. And I don't know the statistics on that. As far as I understand, evolutionary models hold that due to a combination of the various selection pressures and these mutation rates, and that it's a parallel processing and not a linear processing event. And now I'm speaking computer language again. Sorry. How would you say that in genetics? Sorry, a parallel processing versus linear processing. How would you go about to explain that?
Linear processing versus
Parallel
processing
in genetics. How would you explain that best?
I actually don't know the terms. We have like different lineages, and then there's vertical descent, and then diversification. I just want to cut to the chase a little bit.
Yeah.
You're actually, and also for your sake and the audience, you've actually taught me a few things I didn't know just in this discussion. And I'm not going to pretend that I can answer you adequately based on the genetics itself. And that's why I was quick to say, I'm just going to assume you have a very good point.
And then I have to be quick and say, I'm a layman. This is my understanding of other people on YouTube.
So I'm going to totally grant that, and the young Earth creationists hate me for saying stuff like that. But I said this is not easy stuff, guys, and let's wait a little longer. There's still a lot we don't know. It'll start to come clear, and also like you're talking about the mutation rates and all. There's a lot about mutations of DNA elements we don't know. It's not a consistent rate across every segment of DNA. And there's there really is a lot we don't know, not to mention, a lot of people don't know this, our sequencing capabilities are not that as strong as we think. So just for this benefit of the viewers, when we do a DNA sequence, because of the chemistry of DNA, it just fragments into bazillion pieces. So you take a chromosome that has 50 million nucleotides in it, it's going to break apart into segments that are like only 100 nucleotides big. Then you put it in a sequencer and you try to reconstruct the way that whole chromosome is put together. It can be missequenced. And there's been a persistent problem that we don't absolutely know what's actually there. And no one wants to talk about that. And some of the creationists have rightly so expressed frustration. They said, if we could only get better sequencing technology, we're certainly trying our best, but it could be far off. So I'm going to just have to say that I will accept that your prediction has been fulfilled. And that's why I did the detour on the bent pencils and geocentrism, that it does look evolved. And I'm gonna, if I could share a slide from my presentation at CRS, and I told the audience, I said, okay, I was arguing that there's no universal common ancestor of the protein/genes. And even Dr. Dan agreed with me. But I said, conversely, it does look like common ancestry from an organism. And I showed this diagram at the talk. And I said, okay, on the left is hang on, let me I have to remediate something. If you'll excuse me. Just so you could see the diagram, one moment. On the left is human zinc finger, on the right is the pig's zinc finger. I highlighted in red and purple the salient characteristics that define this particular zinc finger 136. And you could do this across all these species, and you will build an evolutionary tree they call a phylogenetic tree. And this would argue very powerfully for evolution. And what I tried to tell the audience, I said, you have to be really careful about these arguments. I was able to definitively prove argue that there's no universal common ancestor gene or protein. And that looks all well and good. But then when you look at individual genes and their trees, this looks like to fulfill an evolutionary. This is consistent with evolutionary predictions. And I'm just like, okay. And then what I went on to do is I said, now consider the problem of the bent pencil and geocentrism, and how this relates to my professional work. A little bit of self-promotion. Since you're in computation, and there are a lot of guys watching this who are in computational stuff, I have said that the patterns of similarity and diversity are optimized to help us understand human biology. That is to say, every creature has pieces of the puzzle of how human biology works. So when we want to study human neurology, we study these roundworms, these C. elegans or nematodes or whatever they're called. And if you want to study nerve, the central nervous system and brain function. If you want to study individual nerves, we'll study squids. We understand things about human DNA by studying yeast, and then translation by studying bacteria. And there's even things like introns that are in plants to understand human introns. And then the chimpanzees and mice are so close to us, they're really great model organisms to help us really understand biology. If we did not have these creatures, we would have to be resorting to dissecting other human beings. This is a fetal pig. And I told this to the creationists. I said, you do have some patterns of similarity and diversity that are consistent with what evolutionary theory would predict. But so is the sun rising and setting every day consistent with geocentrism. It doesn't make geocentrism correct. What you have to look for is an anomaly that will break it. And I said, that is young Earth creationism. If you show that the Earth is young, that's the anomaly that will kind of be the deciding, that will decide between old Earth creationists and young Earth creationists. Now, to go back to what I'm trying to go to here, and this is related to my professional work and a paper that is under review. There are people that have already pioneered this technology, so this is not, what we're adding in our paper is not especially new. But if you take the sequence of, say, a collagen in a human and then a collagen in a zebrafish, and you get other creatures in the mix, and you put it through a computation known as DCA, direct coupling analysis, or AlphaFold, you put it through that, it'll pump out the three-dimensional, it'll give you the XYZ coordinates of the shape of the protein. That's like voodoo. The patterns, see, this is similar, the human is similar to the zebrafish, but you put it through a computation that looks like this. You can find this in Wikipedia called direct coupling analysis. It's a brutal, miserable algorithm. You put it through a few more computations, it's going to make this an XYZ coordinate system that you could predict. And let me see if I could just remove this now. So we've said then, as we are looking at, and since you were mentioning the mutations and the differences and the diversity, we as creationists would say that was ordained by God to help us make patterns like that so we could do scientific discovery. It's very hard to actually see what a protein, how it's shaped. But when we can use other creatures and their sequences, pump it through that direct coupling analysis computation, we can actually visualize the way it's actually shaped. And there's no reason from chemical principles that patterns like that should emerge. And evolutionists will say that naturally happens with evolution. And that is a long-term project of mine to say, no, that's not consistent with random mutation. It would have to be very specially designed to be kind of a self-decoding system like that. And so I know I'm not really giving you very good answers, and I'm saluting you for, even as a layman, what you've come up with. And that's really the best I can give you. So
Sal, I'm kind of frustrated right now. I mean, look, all this information is great, but like I'm confused about what your response is to Miklas's prediction, because on one hand you said like that's a good thing and I'll grant it to you, or something like it's a good argument and I'll grant it to you, and I think the observations do lead there. But then you also said that we don't even know if those are mutations. So I'm confused about like if you're like think he's making a good prediction or not. If we don't know if those are mutations, how can we possibly say that that's a good prediction?
Can I
and
try?
I think, yeah. Well, I know it may be frustrating to hear me talk like that, but a lot of the proof texts, a lot of the proof methods is we even grant certain things, even for the sake of argument. And I personally do think Michlas made a good argument.
Can you tell me what the argument is and what
Okay,
you
so
think
let's
is good?
assume that let's grant that our understanding of mutations is correct. With that, I'm saying that he's talking about the mutations and the differences. I'm going to even grant that, and I'll say that that's good because, and the reason I'm granting it is because of patterns. Let me share my screen again. Anytime you're able to generate phylogenetic trees, so let me share this again. Anytime you can generate phylogenetic trees of where you relate from current slide. Sorry. Anytime we can create tree-like structures, like this, where we sample various organisms, or like say we just sample all the humans, and then we can build trees like this, we can make certain assumptions about the rates of mutation or whatever, and what has happened, and then all the timing of the events. And that's a very powerful argument. So I'm not going to even try to go after that. What I've said is, let's grant everything that Michlas has said, that it's consistent with predictions. And what I countered was, even if it's consistent with evolutionary predictions, it doesn't make evolutionary theory correct. Because geocentrism made tremendous predictions. The observations were consistent with those predictions. It didn't make geocentrism correct because there were anomalies that were not accounted for. You have to look at other data sets to disprove it. You can't focus on one data set. You have to look at the whole picture, and then you see that it's wrong. Because if you focus just on this topic, it ends up not intentionally, but it ends up inadvertently becoming cherry picking. You have to look at other data sets to falsify it. I don't know that I'm getting through, but
No, I would say I agree with what you have said so far about that, and I appreciate it. So if I could quickly reiterate what I think I hear you say is that it's a fine data point, and it seems to be, if we wanted to control it, a valid point that could be used as an indicator, but only an indicator. And basically, you could try to build the house of this. But if someone can remove the entire foundation by saying the Earth is 6,000 years old, somehow prove that, or something else like that, then it doesn't matter because we have suddenly shown that, yeah, the Earth is not the center of the universe, basically. Or in this case, that sure,
Right.
it
Right.
really does look like it conforms with evolution. The prediction is fair, it's good. But if evolution is impossible due to something else, then there must be some other reasons that it conforms with evolution.
Yeah, so this is why I keep emphasizing geocentrism and the bent pencil. I keep telling the creationists it does look evolved. They hate me saying this. I said it does look evolved, just like it looks like the world is geocentric. Okay, you can't, and because I deal with, I've gone through school and I was a theistic evolutionist, old earth creationist. If you look at isolated data points, you would conclude that it looks evolved. And I wish creationists would just admit it and say it looks evolved if you look at a narrow data set. The way to resolve the problem is to look at the wider data set that doesn't deal with genetics at all. It deals with physics of the solar system. Then you have a different angle at this. And then the question is, then why would God make these patterns? Is it to deceive us? And I've tried to say, no, it's actually optimized for scientific discovery. You just have to kind of look at it in a different way. And I know that's frustrating. And the creationists are like, oh, we're going to just win every argument. I'm like, no, you're not. The heliocentrists didn't win every argument over the geocentrists. They had to appeal to data sets outside of those narrow points. And I'm just like, creationists, quit arguing like that. You're going to lose. And I know that's harsh, but it's like, I've been through a secular curriculum and I'm trying to just, alert people. It's like, this is what you're going to face. And it's not as cut and dry as you'd like to think. And you don't serve our cause very well by pretending it's otherwise. So don't look so sad, Rebekah. I didn't mean to just
No.
Oh.
Sal, I'm not reacting. Like, I'm all the information that you're saying is good. It's just I'm trying to get an answer to a specific question, and I don't feel either that you're hearing the question or that you're like responding directly to it.
And I'm not responding directly to it. You're absolutely right. I changed it for a reason, and I'm saying this is not the way you're going to win the argument. You have to change the foundation.
But I still want the answer to the question. Like, I still want the answer. Like, I don't care which side it's going to be good for. I want the answer to the question.
I think, okay, I gave you my opinion.
Do you know? I want to ask the question. As I asked it to Dr. Dan, I sent him this question this morning after I rewatched me and Michlas's thing, okay? Because I was like, you know what? I need to know, do we have observations that back up Michlas, or that like would show Michlas's prediction, which way it favors? Here's the question that I asked Dr. Dan. He hasn't responded yet. That I said, do ERVs that are shared by more organisms contain more mutations than ERVs that are shared by few organisms?
Can you repeat that again? I'm sorry.
And Michlas, am I asking the right question here?
That would be the right question for the evolutionary side, yes.
Okay. So do ERVs that are shared by more organisms contain more mutations than ERVs that are shared by few organisms?
I think the answer is yes. And so you wanted to know what my viewpoint is? I said, I'll side with Michlas on this, that it's consistent with evolutionary predictions. That his viewpoint is consistent with evolutionary predictions. I'm sorry to burst your bubble.
No, you're not. I don't have a bubble to be burst. I just wanted an answer to the question. There's no bubble. It's just I'm just wanted the answer. So which
right.
observations have we made? Like where have we made these observations? Which experiments have we made these observations?
We sampled ERVs across a large number of species, and it would look like those phylogenetic trees I was showing with like the proteins of collagen. This is very consistent across all proteins, and I expect it to be the same with ERVs. We see this tree. You keep hearing about the nested hierarchy. Biology, that nested hierarchy pattern is very, very, very strong. Creationists said it's not there. I'm just like, I look at gene sequences all day. It's definitely there. So let me show this here. And I'm sorry I don't have an ERV diagram because I'm not a virologist and I actually don't like doing genetics. I like proteins. So, but let me show this protein diagram again. And
while you find it, I can say I also afterwards talked with Rebekah, asked some people here among Dr. Dan, and I got 3 different answers from 3 different people who did send me some papers that I only scantily understand, to be fair, and they did seem to hint at an agreement. But as far as I could read, it had not been directly tested, tried, specifically like I said it. So I can say it seems like it's that direction, but it's not determining, at least not those papers that I read and what I could understand from them.
And I guess then my question is, when we're considering like the great differences in amounts of time, we probably need to consider first the idea of whether or not these are actually mutations or are they shared, like were they already there to begin with? And that's something I don't know. I mean, is there any way to figure out if something is a mutation?
No.
So,
not very well. Okay, so let me back up a little bit, and I'm going to give you the correct terminology just to help you.
Thank you.
I'm sorry to. So organisms that share, if organisms share the same ERV, that is, they're homologous, but all the homologs exist only in one small set, we would expect them to not be very diverse. Whereas if we have homologs that are in a large set of creatures, we would expect it to be very diverse. That definitely is consistent with evolutionary theory. It would be a prediction that is satisfied. But that's why, and I know it's frustrating. You said, Sal, you didn't answer my question. I said, just accept it for the sake of argument that it's true. Because I just keep trying to tell creationists, stop trying to nitpick here because you're going to lose, because it is consistent. This is not the battlefield to win it on if you're a young earth creationist. You have to look at wider, at other data sets. This is not sufficient, this is not sufficient to rule one way or the other.
But I mean, this is, and go ahead, Michlas, go ahead.
So you asked if there were any ways that we could see if there were mutations. I had hoped that there were a way that I had thought about, and I asked about it. And sadly, it seems it is a way, but it's very limited. And that was to take the ERVs that we find across multiple lineages, and then compare all of those ERVs that we do find with actual living viruses and see are any of these viable. The problem is that we cannot move, what would you call that, many nodes back in the so-called tree of life before the ERVs that we do have are so deteriorated that we would not be able to compare them that directly with a living virus, as far as I know. Because my idea was then see, okay, what are the mutations compared to the actual virus, and then say, okay, those must be mutations. Because even if God made the ERV as a what was the word? I can't remember what Donnie said, but as some sort of mirror to the virus to fight the virus, well, then it must look as much as the virus as possible. But sadly, it seems it's too deteriorated to do that, and I don't know if there's other methods.
I'm going to give you, okay, Rebekah, I'm not trying to be, I try to be a straight shooter, and the straightest shot I gave you is just assume he's right. It is consistent with predictions, but it doesn't mean that the theory is correct.
Yeah, I know. Believe me, you think I'm like emotionally involved with this or looking for some knockdown argument for young Earth creation. I'm not doing that at all. I'm just trying to figure out what the facts are.
All right. We cannot actually determine if they're mutations or created. All right. And that.
Okay.
If we prove, we can determine that it was created if we prove the Earth is young, because there's not enough time for it to mutate. We know that there are limits to so the DNA replication process has high enough fidelity. We can estimate, that it's not going to mutate faster than a certain rate.
Okay.
That it had, and so if the Earth is young, then we would know that the differences were created. That's how we would know. Everything hinges on the age of the Earth. If we assume that all this is old, I don't know that there's any way, because like the progressive creationists and they're, godly Christian men who are progressive creationists, that God would just create over time these new ERVs, and maybe they diversify. I don't know that there's any way I could uncoil that and say, well, did God just create it like last Thursday, or did he create it deep in time and then it diversified? The only thing I could say is that if the Earth is young, say less than 10,000 years, then definitely all those ERVs, we can kind of make a guess that they were created in place as they are. And I'm going to point something out really powerful for you.
Okay.
And it has to deal with zinc finger proteins. Did anyone talk about zinc finger proteins? Because this relates to the ERVs. And that's
but hold on. Don't go to another topic until because I really want to get to the meat of this topic and say, based on what you're saying, then since we cannot know whether something is a mutation or if it's created, then Michlas's prediction is it is irrelevant to whether it cannot be actually predictive, because it's circular. Because it's like, then if it's one case, then we'll see more mutations, but it assumes that those are mutations.
I would say not circular, it's indeterminate. You need other data points to resolve, because you could have two ways that that you can interpret the data. You need to change the topic so you can determine what is the correct viewpoint. And I'm not trying to, rather than say circular, I would say it's indeterminate. It hinges on data outside what we're looking at.
Okay.
Yes. So basically what I get from you is either we would need a way to establish that it is mutations. So that would be, for instance, like the one I had hoped for, but that just doesn't work. But if we had something like that, or we need to prove that the Earth is young, because those two things would falsify the opposite position.
But there's another way, and it involves the zinc finger protein.
Okay.
The easy, cheesy way is prove young Earth. And Dr. Sanford said, can you prove this some other way? I'm just like, thanks, boss. And it's the zinc finger protein. All right.
I'm writing this down. Zinc finger protein. Tell me all about it.
Okay. I had a naughty beat. I posted something naughty on the internet. I said, giving evolutionary biologists the finger. And it was the zinc finger. It deals, okay. So we can do a show on zinc fingers, and I don't know how long you all have time for this. But again, all right, so rather than saying circular reasoning, say it's indeterminate. We had a valid point. How do we know that it's created versus evolved? And I said, the easy, cheesy way is if the Earth is young, this settles it for the most part. But if you have an indeterminate system, you have to appeal to other data points maybe to resolve which way the truth falls. The more immediate thing available to us is zinc finger proteins and the CAP1 complex on ERVs. And no one talks about that except me.
Okay.
And I said, this is giving evolutionary biologists the finger. And I gave them fits over this.
Sal, we have to do that on examining evolution. That would be so funny.
And okay, so I gave this presentation to a bunch like 40 Christian faculty who are biologists, biochemists. And they started laughing at the end. I said, look what the evolutionists believe. And they were laughing. They said, well done, Sal. Okay, so this is part of my professional work with Dr. Sanford. And it is nerdy, but it's from an angle no one else thinks about. Okay, so let me just show the architecture of the zinc finger protein. And let me see if I could show it here. Now I didn't have, so first off, we're gonna, how about we go ahead and do that, schedule it to get into the details. And I'm just gonna a cursory exploration of this because I don't have the slides immediately handy. So Michlas, I would very much invite you to watch the show I do with Rebekah, because this is a professional presentation I gave to Christian faculty who teach both at secular and Christian schools. They're biology faculty. I can't BS them, all right? They would shoot me down. This is not like the internet. And I was very flattered. One of the deans of the university said, "Can I share that with my biology students?" I was like, "Holy smokes! This is prime time already." Okay, so let me show you the. I'm going to show a number of screens. This is going to be a clunky presentation because I don't have all the slides handy. So the genes code for proteins. They make amino acids, and the amino acids are these long strings. You could represent each amino acid by alphabetic letters. And let me just zoom in. This is zinc finger 136. And it's 540 or so amino acids long. And there is a distinctive pattern of the zinc fingers here. So each row, and please interrupt me now as I walk through this, each row represents a zinc finger domain. And let me see if I have each row represents a zinc finger domain. What'll happen is, as this protein is folded, a zinc ion will connect to these two cysteines and these two histidines. And then another ion will connect here. And that's very important for creating the shape of the protein. So now this is where it gets a little clunky. I'm going to have to Google around to find pictures of the actual fold. So you have like this, this is a picture of the zinc finger here. You have the zinc ion here, and each of these domains. Let me see if I could find a zinc finger array. Zinc finger array. And I know Rebekah gets kind of impatient when I don't get right to the point, but I have to talk about so many things so you can understand. So let me see if I have, okay, spelling. Let me see if I can find one. A little better. Zinc finger array. Oh, I know what. Zinc finger CAP1 complex. The chase here. Yeah, baby. Got it. All right. So here is the DNA. You see that kind of squiggly thing? And the DNA wraps around histones. You remember chromatin, Rebekah? In eukaryotic organisms, DNA is wander around these little spools that we call histones, and the histone and the DNA complex is called chromatin. Here we have a zinc finger protein connecting to an ERV. All right, so when you have like passwords that match logins, or you have keys that go into locks, the spelling of this zinc finger has to match the DNA. It has to match the DNA. So let me show this again here from current slide. The way this is spelled makes it match that sequence of DNA. And the reason you need so many of these is so that it will connect to the right location of DNA, as we see here. Now, why am I pointing this out? For the ERV to function, what it does is this zinc finger connects to the right location on the ERV. The zinc finger then connects to other proteins to make this molecular machine. So the ERV is essentially a parking lot or a scaffold for that specific molecular machine. And that, I'm sorry?
You say for the ERV
yes.
to function?
Yeah, for the ERV we're finding one of the functions of the ERV is it serves as a specific parking lot, a scaffold. It's a specific parking lot. The sequence of DNA is very important so that this particular zinc finger will know to park there. When it parks there, then these other proteins will come to that zinc finger, and it has to have all these matching interlocking parts. They have to be spelled just right. And then this machine will assemble in the right location, and then it modifies these histones, these chromatin, they call it chromatin modifications. That's very important to the functioning and regulation of DNA and expression. There's a computational system here that is involved. So this cannot evolve randomly, because if the ERV evolves randomly, then the zinc finger that's supposed to connect to it and make it do all this regulation will just be floating around and not being able to help assemble the machines in the right location. And so what ended up happening, and the reason the faculty were laughing when I pointed this out, the evolutionist said, look at all the coevolution that happened. And I'm just like, that doesn't look random to me. So save this for another show. I'm not doing this justice. This is like a 1.5 hour presentation. But the point is, you can't have random mutation. You can't have random ERVs just coming out there, and then this wonderful Cap1 zinc finger complex that does all this computational regulation and expect it to work. It has to be created. So if we're going to argue an old Earth, we'll have to say God made progressive creation, and we had these things miraculously created in place at a certain time in our history. And there are progressive creationists. So if we didn't want to settle this in terms of young Earth versus old Earth, we can assume old Earth with miracles along the way. And this is one of the miracles. And that's why I said, for me, this is giving evolutionary biologists the finger. And that'll be, I think you should make that the title on examining evolution.
Oh yeah, that was my plan. That was my plan.
All right.
Yeah.
Okay.
See, okay.
I just got to say, Rebekah, you get me in a good mood, because you saw me backstage. I was pretty depressed because I'm in the middle of, a lawsuit against my church. You make me so happy. I could talk about stuff like this. It puts a smile on my face, and I can talk about giving evolutionary biologists the finger. And you just made my day that you said you're going to make that the title on examining evolution. I can bet we're going to get a huge audience. So there you go.
Yeah, I wanted to bring up, if it's okay, is Michlas, you mentioned it before, one of the things that Donnie talked about ERVs, and you responded to it, and now I kind of want to give a little bit of a response to your response.
Yes, of course.
So Donnie said, this is a little bit strange that, because we've found that some of the functions of ERVs is in fighting off viruses. So Donnie's like, hey, this makes sense that it is, the sequence of it is like a virus, because then, the function of it is to fight off viruses. So it makes sense. And he said the evolutionary explanation is that these were originally viruses outside, came inside, and it's similar to and then started working for our bodies, right? And it's similar to like a robber breaking in your house and then turning around and being the security guard. And you responded to that, and you said you don't think that's a good analogy. You said it's more like a technically superior nation with special forces that you defeat, and you take their weapons, like somebody, a force attacks you, you're somehow able to overpower them. You take their weapons and then you use their weapons against them. Okay. So that was an interesting analogy, but what I notice about it is that there's an agent there. There's foresight. There's intelligence involved, right? So you take the weapons and you use them against them. That takes, some type of knowledge, right? And that is what the cells don't have, right? We don't know of any thing within the cell that would give it such foresight to be able to, take a weapon and turn it and use it in a different way.
I don't know how it came about, but as far as I know, we have multiple immune responses and immune cells that does exactly that.
Right, but
picks apart things and then looks against it. So how it came about, I don't know.
And again, the immune system's another good example that I think you should look at that and say, that is not something that can evolve. That takes foresight. That takes, so if you're citing the immune system as an example, again, you said you don't know how that came about, but you're kind of assuming that it came about by natural processes and using it as an example, but we don't know that it came about by natural processes.
We don't know that it came about. No, of course we don't know. No, well, I don't know. No, that it's wrong to say we, because I can't speak for all biologists out there and so on. But compared with how adaptive and coevolution in general works, I don't, on the face of it, see the problem, but there might be one, I don't know. I don't on the face see how that seems improbable, to be honest.
Well, and again, this again, if you start with the assumption that evolution happened, then of course it seems probable. But if you start from a neutral position and say, I don't know how things came about, and you consider what it would take for something like that to come about, it seems to me that it would take some foresight, some planning, some awareness of the attacks that might come toward the body, and a planned way of, fighting that off. And I don't see where that foresight would come from in the evolutionary process. So for me, that is one of the main reasons that evolution is a preposterous theory, is because there's no explanation for the seeming foresight that is present throughout, not just in the immune system, but in so many systems in organisms.
Can I add just one thing mechanically here, Rebekah, to your point?
Sure.
So this is the PubMed. There's an article on the CAP1 regulates endogenous retrovirus in adult human cells and contributes to innate immune control. What I was trying to illustrate with the zinc finger, for this CAP1 to do its regulation, it has to have all this machinery, including the zinc finger. Where the foresight is needed is to get the spelling of both the crab zinc finger sequence, and you saw those alphabetic letters, and it has to match the DNA. Now this is where it gets into, this is like matching passwords for a login. You can't, you know, when you're trying to make the parts fit for a system, you have to have foresight that you're going to spell things a certain way, that it attaches only to this and not to something else. Otherwise, the system will fail. What'll happen is, you know, if this is spelled wrong, it's going to park in the wrong location and it's not going to do regulation. Because like with a lot of mechanical systems, it has to be positioned in the right place to be able to perform its tasks. And
could I request one thing, Sal?
Sure.
For the future talk that you're going to have with Rebekah beforehand, and this is one thing where I don't know the answer again, because there are many of those.
Sure.
But could you examine what are the various proposed origins of the first retroviruses? Not the ERVs, but the first retroviruses. The evolutionary models. What are the suggestions that you have there, unless you already know it, of course.
Okay, this is a good Dr. Dan question. And we had a discussion on that. We think all viruses, there's a good argument to be made that all viruses ultimately started with cellular organisms. Viruses didn't come first, cells did.
So they broke out from a cellular organism.
Yeah.
When did the zinc finger originate?
The zinc finger originated in like vertebrates, around the vertebrate time, maybe a little earlier.
Okay, so there's no zinc fingers earlier.
The eukaryotic zinc finger emerged. There are prokaryotic zinc fingers we found out recently. The eukaryotic zinc finger emerged, you know, like I think around the time of vertebrates.
Okay.
Someone can check. They're not
those
very...
zinc fingers, zinc fingers, sorry, are made so they match the DNA of the prokaryote.
Of the eukaryotic ERV,
no,
but I need
the original ones. You said we had the zinc fingers in prokaryotes,
okay.
Right?
There are prokaryotic zinc fingers. I need to point out something because this can be a little confusing. Zinc fingers can attach to anywhere in the DNA that it's spelled. It can attach to genes, promoter regions, all sorts of DNA. It doesn't specifically attach only to ERVs.
No,
it attaches to something that's spelled a certain way.
I get that.
Okay.
But if you had retroviruses originating from prokaryotes and you had zinc fingers in those prokaryotes, that means the retroviruses would already have a somewhat matching key. The first retroviruses would have a somewhat matching key to the first zinc fingers. And that gives them a path for co-evolution because they are already aligned. They don't have to match everything from scratch.
The architecture of the zinc finger in a prokaryote is very different than the eukaryote. It's like we can build engines with carburetors or fuel injection, but everything else is very similar. This would be akin to it because the way that eukaryotes implement the zinc finger is very radically different than the way prokaryotes and eukaryotes implement their zinc fingers differently. It can't just be imported from one to the other. But you do raise an interesting point. Were there retroviruses hypothetically before zinc fingers? And we could grant that. What I would just say is that we might have certain ERVs that look created.
It would be more interesting if the retroviruses came after the zinc fingers. If they came out of cells from creatures that already had zinc fingers, then they had the partial key.
Yeah.
Right.
So maybe if we have that talk on Rebekah's channel, which will actually be spending a lot of time just describing all the cellular architecture, and it's going to get into the weeds. But then the reason I like doing things like that is everything's a little bit clearer. And then also it gives me a chance to say, we're looking at this way too simplistically. When I'm seeing these arguments, I'm just like, they're data points that have emerged only like in the last few years. And I'm seeing kind of old arguments on both sides. And some of the cutting edge is just not really well known in the YouTube sphere. I'll talk about it in my nerd torture sessions, but then it just, people, it's like, that's just too painful to sit through. It's like, I get that,
And the problem is, I like them, but I just don't have enough knowledge to follow, basically.
Well, if you want to join on that one, Rebekah, I mean, I'm okay with Michlas joining us. It would be just a matter of coordinating it between our three different time zones.
I think coordinate between the two of you, then tell me what time you have figured out, and if I can, I would like to be there, but don't match it for me.
Sounds good.
So I have the slides ready that I made, these presentations 3 years ago in university, to university faculty. I just have to dig them up, and we can walk through it. And one reason I enjoy having you as as kind of the host of the show is you can help clarify to the audience, when I start to go off in things where I'm using specialized terms, you can help me say, okay, pause, Sal, can you explain that a little more? So I think you'll have a different view of ERVs after we have that presentation.
Cool. Thank you. Was there anything else you wanted to discuss, Michlas?
I think we've been going on for so long, so maybe it's not the time right now for that.
There was a question in the chat earlier that I was hoping you would address, Sal, because it's something that came up in my Bread of Life stream yesterday. It's from Assembly of God, and he said, what about the zircon crystals? This is like a knockdown basically against young earth creation. And what's weird to me, the reason I'm really, really interested to hear the answer to this is because I recall from my early days of young earth creationism that the zircon crystals were one thing that were like promoted as good evidence for the young earth. And so now, yesterday and now in this stream, it's been presented that the zircon crystals are against the young earth, but I don't remember what it was that it was like support for young earth. I think it had to do with like the helium that was still in the zircon crystals or something like that.
Yeah, I haven't followed that in a long time. So Assembly of God, I'm sorry, I don't have a good answer, but it used to be that exactly as Rebekah said. So I think, as I said, young earth creationism is hard. ID is easy. There's just so many data points that are argued well on both sides between old earth creationists and young earth creationists. And I think we just have to wait on the Lord to give us the data points to settle it from a scientific standpoint. I accept I accept young earth creationism as a matter of faith, and some people may have an objection to that. And it's like, okay, so we'll just see how this plays out. And thank you for the kind words. He said, okay, Sal, you're awesome. So this young earth creationism is way harder than I think young earth creationists tend to trivialize and say, oh, look at our few data points, we won the argument. It's like, please don't do that. This is a tough fight,
Well, one thing I feel like, when I tell people there's some evidence for young earth, they usually slam back, there's no evidence for young earth. And I admit that there's evidence against young earth, but then I say, but there's some evidence for it. And I think what's happening here is that like, it's kind of how the media, how they like spin stuff and like they select certain news. And in the same way, like certain news is widely broadcast and other news is not broadcast. And that's a way of like not, like of favoring one version of events, right? Is like not putting all the news out there. And so I feel like, when something points toward a young Earth, it's quickly brushed under the rug, and like no one talks about it, and everyone just, oh well, that's a, like the dinosaur, the soft material, the biomaterial, and go ahead.
I wanted to say part of the reason both sides do this is people want to believe that we figured it out, and also these organizations do solicit donations, so they, no one's really eager to say, oh, we really don't know. No one wants to hear that usually, but sometimes you have to tell the truth and say we haven't settled it, but they'd prefer to say we're settled, you're comfortable, a-okay, and that'll get the donations in. And that's what, people like resolution, and I get that. But what holds me from saying that is, when I think there are arguments on both sides. I will point out in the debate with PhD Tony, what he didn't challenge was the magnetic field of the Earth. That is a testable prediction. And I even pointed out in that debate on the Wikipedia entry, the testable prediction is the magnetic field will decay in 1600 years. Now, we won't be most likely around to see it, but civilization can actually see that prediction take place. That will affect climate, because the Earth magnetic field protects us from the solar wind, which could strip away our atmosphere and also the ozone layer. So that is a testable prediction. And I was like, okay, I saw what Tony would attack. He would attack the faint young sun paradox. He didn't. He tried to dismiss as irrelevant the chemical dates, and I could have been a little bit more forceful and say, no, you can't just dismiss it. It's problematic for your side. And I know other geochronologists that know that, yeah, that is a problem, Sal, for old Earth. And he didn't even touch the magnetic field. And I'm like, okay, you gave me the data point I needed. So if I were going to give some order to things of arguments in favor of young Earth creationism, PhD Tony kind of gave the game plan. The magnetic field, next is the chemical dating. It's going to be a harder fight on faint young sun paradox. And in his favor is the consistency, the concordance of radiometric dates with other things, and he made a very good argument. So sorry I had to go on that tangent, but I know some people are wanting to hear, I just had to, when people say there's no evidence, I'm giving you three major evidences. And like I said, I said in my debate, a single ugly fact can bring down a beautiful theory. So right now, this is the battle of ugly facts. He has radiometric dating, and I have the magnetic field and chemical dating.
Cool. Well, thank you so much, guys. This has been great.
Yeah, thank you. It's been interesting, and I will look forward to at least seeing your talk, and hopefully be there at the same time.
And
Okay.
Lindsay from Secure the Harvest is premiering a video right now, and she's really just starting out. It would be great if anybody could go over and subscribe to her. And I posted the link. Let me post it again.
Yes, please.
Here's the link if anybody wants to go. This is the video she's premiering right now. And so, yeah, she's got a lot of good Bible study going on, so please do subscribe. Thank you.
Oh, I forgot. I'm the administrator of the channel. I was waiting for you to end the discussion. I'm sorry. All right, so we'll wrap it up. I'll meet you briefly backstage to just say goodbye and talk about any future things. So thank you, everyone, for joining us. Take care, and God bless you.
Yes,
God bless you.
Okay.